All right, you guys, let me pause this real. All right, here we go. Sorry, I had to quickly do a couple things. Anyways, so you guys, what we're going to do is do a quick little lecture. What I like to do here, let me go back to this. So right now you're in transcription, so we're learning about a specific one step into how our cells make proteins. And what you'll notice is I'm giving you some options here. You've chosen option 2. You do not need to do option 1 or option 3 unless you want to. Okay. And so anyways, to do this, let me just quickly get started, and I apologize for the sound. It's the joys of having to work at home, I guess. So you guys, to first start out when we're looking at this, what we're going to do is we're going to look at a new molecule called RNA, describe how RNA differs from DNA. So you'll be able to do that after this lecture. You'll also be able to explain how the cell makes RNA specifically and what role that plays in how the cell makes protein. And so to do that, you guys will quickly, let's just look at this. So when we're looking at DNA versus RNA, you guys know the structure of DNA. We were able to do that in class before you guys left. We know it's a double helix. We know it's made of 2 strands and that the strands combine together in between by hydrogen bonds in between these 2 bases. We know that in DNA, A binds to T and G binds to C, right? And we know that when it does, it's going to form this kind of twisted ladder structure after it's done. We also know a couple other things. So we know that DNA, it's got on the backbone, these little rungs, or not the rungs, but the legs of these things are made up of it's alternating phosphate sugar, or sugar phosphate, whatever way you're looking at that. And we know that the specific type of sugar that it contains is called deoxyribose. Okay. When we're looking at RNA on the other hand, though, RNA is single-stranded. Okay, it does not form a double-stranded helical structure like this. It actually will stay single-stranded like this. It does sometimes form and bind to other RNA molecules, but it will form. It's fine being single-stranded. The other unique thing about RNA is that it's got A, C, and G, but it does not have thymine. Thymine is unique to DNA. It actually has a different one that is in replace of thymine. It's called uracil, or U. So in RNA, G will bind to C and A would bind to U if you had 2 strands of RNA here. Another interesting change is that the sugar, it's not deoxyribose, it's actually ribose. And that's actually where the name comes from. So ribonucleic acid, this is deoxyribonucleic acid. So that's some key differences when we're looking at DNA and RNA. So what I would recommend you guys do, since you are taking you decide to watch the lecture, take some notes of this because there are questions at the end you would need to answer. Another thing that I want to point out is that if you are still struggling with these concepts, if you want to, you can get the online version of the text and you can go to chapter 14, and I believe it's section 1. Okay, so chapter 14, section 1, page 440 is where it starts, okay? And it will go over some of this information as well. So anyways, when we're looking at that, I kind of just talked about those key major differences there. You can pause the video whenever you would like if you need to write down these notes, but I'm going to move forward. So when we're looking at RNA, there's actually 3 main types of RNA. The first one is called messenger RNA. Another way we kind of model that is we kind of label it mRNA. And so where are my tools here? Anyways, interesting. So mRNA is one that we have, and then we have ribosomal RNA. We would call that rRNA. And then we have transfer RNA, which is tRNA. And so let's look at these specifically. So messenger RNA, it's really important structure. Messenger RNA is what makes a copy of that DNA. And I thought I had a picture here. Where is my stuff? Interesting. Okay, well, I guess I don't have a picture. I thought I did. I'm sorry, you guys. So let's just go back, I guess, here. I do want my tools. Where are my tools at? Anyways. What does that do? No, it's not what I want. What does this do? Got it. No, I don't want that either. No, I don't want subtitles. Block. Okay, here we go. We'll just stop messing. And that probably just ends it. Okay, so when we're looking at this, you guys, so mRNA is a molecule. It's actually made from DNA, and so what actually will happen is DNA will open up, and it will take a section of that, a portion of that DNA, and make like a gene, and it'll actually make a copy of that DNA. And the reason why it does that is DNA does not want to leave the nucleus; it wants to stay in there, but mRNA can leave the nucleus and it can go to other parts in the cell where it can then be made into a protein. And so that's kind of what it talks about here, is that after it leaves the nucleus, the mRNA will travel to the cytoplasm, which is wrong. I mean, it's in the cytoplasm, yes, but it goes to what we call the cytoprotein synthesis, and we're just going to put the ribosome. Okay, so we know the ribosome is where protein synthesis occurs. Okay. All right. So ribosomal RNA is another type of RNA there. And what it does is it actually is what makes up the ribosome. So ribosome is made up of two subunits, and it's actually got some protein involved there. But anyways, these subunits consist of RNA. All right. Transfer RNA, so that tRNA, the other type, these are very important in the later step that you'll learn about later on. But these are specific type of RNA that helps to bring what we call amino acids to that mRNA to make our protein. Amino acids are kind of like the little parts that make up protein. Okay, and so the tRNA will bring the correct amino acid in, and that'll make more sense later on. All right, so here we go. So now let's dive into this, looking at how this is all made. So we're looking at transcription. This is kind of the first step in how proteins are produced. And if you look right here, what we see happen, the first thing that will occur is RNA polymerase will come in after the DNA has been split and will start to create a RNA strand. Okay, so for example, if the DNA is reading A right here, it would bring in a U. If it's reading C, it would bring a G. If it's bringing a G, it'd bring a C, etc. And so this right here is showing that mRNA that's getting produced from the RNA polymerase as it's reading this DNA sequence. Okay, and so we're going to dive into this step a little bit here. Yep, Yep, talked about it. Okay, so RNA synthesis happens at promoters, and so promoters, RNA polymerase will bind to these regions that have specific base sequences, and we learned about this earlier in DNA replication. Okay, but there's specific promoters that the RNA polymerase will read and say, okay, this is where a protein starts, or a gene starts, and it'll start to code that. After that RNA is made, it actually goes through some different things. And one of the key things that it goes through is what we call RNA editing. And so this right here is representing an mRNA. We call it a pre-mRNA because it's not fully mRNA. And if you look right here, what it's actually doing is it's splicing out some key parts. These key parts that are spliced out are called introns, while these that are left in are called exons. So the idea is, what is actually being coded for the protein? Is it the introns or the exons? The correct answer is it's the introns. Okay, so we have our our exons that are left in. Introns are cut out. The exons go to be read at the ribosome to be made into the or to be used to make the protein. And I will link a couple reading things, you guys, to kind of for extra help if you need to on this step. Okay, Khan Academy does a pretty good job there, so I'll link a Khan Academy for this RNA editing. So you can see right there one more time, our pre-mRNA, after it's read from the DNA, has both what we call exons and introns embedded throughout. However, it's a waste of time to code this entire thing, and so it's going to split some of that stuff out so that the ribosome only has to make that much, read that much. All right. So from that lecture, you guys, you should be able to answer some of those key questions. And like I said, I'm going to link some assignments there with you for you to read in addition to this lecture for option number 2. And then what you guys will be able to do is answer these questions that are found right here. There's 11 of them. Okay? Alrighty. Let's see. Yeah. And again, if you need to read part of the textbook, you can as well. I just made these different options so that multiple people can, or so if you like the more of the lecture and reading, you can do that. If you want a POGL, you could do that. Or if you want to do more of a hands-on, you could do this. But anyways, I'll talk to you guys later.